The greenery cover on a landmass may seem to be a wild growth of nature's gift in the lenses of the ordinary man. Well, science pries away from that limited vision and studies each plant in the forest for proper scientific profiling. The Plant Genetics Resources Research Institute, PGRRI, located at Bunso in the eastern region of Ghana, is one of the 13 institutes of the Council for Scientific and Industrial Research, CSIR. It investigates all plants, looking out for varieties which are naturally not high-yielding in terms of agricultural produce. The scientists do not discount any crop, as some possess positive traits such as disease resistance and nutrients that may be cross-fertilized to introduce new properties in other varieties. These are the result of careful analysis conducted on plants to map their characteristics for purposes including but not limited to multiplication and distribution of the item for a gain. A crop described by Ghanaian scientists as orphan, in other words, neglected, is the taro. It is called brobe by Akans in Ghana. The tuber produced by the taro plant is the part which is of interest and it is widely consumed. Leaves on the taro plant bear a semblance with contumely, and patronage vary from low consumption to a complete disusage. Relatively unimportant to consumers, it is the leaf that was strenuously observed by Ghanaian scientists to draw conclusions the taro plant was on the verge of extinction. The leaves were blighted, causing the stunt or decay of the taro plant. As the local variety of the crop succumbed to disease, the Plant Genetics Resources Research Institute at Bunso introduced varieties of the crop from a state of the U.S., Hawaii, and from Somoa, a Pacific island, both of which are rigorously defiant to pests and weather. Richard Buampong is the research's focal person on the taro.
We were able to introduce some germplasms from other countries, which were characterized, evaluated, and then in conjunction with Crop Research Institute in Kumasi, we did multi-locational trials and found out that 4 of these germplasms that were brought from outside Ghana were very promising. And for that matter, we decided to multiply them and then give them to farmers to cultivate so that taro may come back to Ghana.
Also under observation in plant regeneration fields are varieties of rice, sweet potato, yam, and cassava. All other plants of interest to the Institute are being cultivated, sometimes under controlled conditions, to be cultured.
For cassava, the stalks are cut in portions like this, and then new shoots will have to start growing, and it becomes a full plant.
At the Plant Genetics Resources Research Institute, the entire cycle of plants from seeds, seedlings, plant tissues to full growth and harvest are subjected to professionally managed storage, artificial germination, and pathology laboratory analysis, where Barnabas Adongo is one of the technicians.
In order to confirm the absence or the presence of the pathogen, we bring these seeds to the lab and do the seed health testing. Apart from that, we also go to the field to inspect the field gene banks. And the field gene banks will mean the crops that have been established at the field, which are normally the recalcitrant ones, those that cannot be stored at the seed store, which are normally at the field, like the cassava, the yam, etc., that are on the field. We inspect them for incidences of disease, and we bring samples of the disease parts of the plant to the lab here for diagnosis.
The director of the PGRRI, Dr. Lawrence Misa Abuaje, who has handled the institute for more than 20 years, believes it can do more in correlation with additional financial assistance.
If we bring a material like Bambara granite, right, and plant it at Bunso here, it will grow vegetatively, but no pots will be sent. So depending on the type of material, of course, in the southern part, in the forest and semi-deciduous forest area, the most important thing is to conserve the tree crops, the tree species. Yeah, that has a long gestation period.
From the raw seeds to processed seeds, as we have seen here in the tissue culture lab, activities represent procedure and culture. They are done to isolate or stamp out disease causal agents before the seeds hit location for regeneration and for the healthy development of tree plantation. Napoleon Atukitu reporting.